Good morning or good afternoon, wherever you are. My name is Duerkop. I'm the co-founder and CEO of Novasign. We're a spin-off focusing on bioprocess modeling. Today, we're going to talk about process modeling and the application of artificial intelligence and digital twins for accelerating bioprocess development. So actually, artificial intelligence is everywhere today. You hear it everywhere from the media, and it's quite clear that this will also migrate to bioprocessing. However, when you think about that, the question is AI a one-hit wonder that will solve all of our problems? And to answer this question, maybe to be a little bit critical here, is I asked one of the chatbots to create a bioprocess for me. So I was quite polite. I said, hey, please be so kind and create an image of a fully continuous bioprocess that connects of 2 bioreactors, each 500 liter, connected to a filtration unit, and then to 4 consecutive chromatography columns. So in order to have such a picture, you know, when you go to a conference, you want to envision this and you want to show people basically how a fully automated continuous bioprocess might look like. And in order to do so, I asked such a chatbot, and the answer that I got was something like that. So what does this mean? From an engineering perspective, I guess you will all agree with me that this is not very, very helpful. However, and this is the important part, what I want to tell you here is that when you don't have domain knowledge and you look at this picture, you might get quite impressed because it looks super fancy and it looks like a very fancy facility that is used to continuously produce a certain drug, obviously. But at the end, if you have domain knowledge, you see that this device or this machine or this facility will not produce anything because it's just simply not a bioprocess like facility. And this is really important. And when you think about process modeling and digital twins and AI need a lot of validation. And especially in our field, when we talk about bioprocessing, this validation is very important. And this is also one of the reasons why we are not there yet. So although we might all envision in the future something like that, we want to have facilities that completely control itself automatically, like you can think about here like an autologous cell therapy facility, where every patient has his own manufacturing process that's completely autonomously driven. Actually, this is where the future, where we want to go. But when we look where we are right now, we are more like something in this domain, a paper batch record-based bioprocessing, where things might go wrong. Operators might look into the bioreactor and say, oh, what's happening here? This is not what I expected it to be. How do we get out here? So actually, there's quite a big difference between Industry 4.0, the vision where we want to go fully continuous automated bioprocesses, and the current reality. And the aim of today's talk is basically to give you a certain idea of how you can master this process from, let's say, Industry 3.0 to Industry 4.0, and what are the important steps on the way. I want to start with a vision. I want to start with the vision of Novasign. The vision of us is to create digital twins, digital bioprocess twins, so representations of your process, basically. A fully representation from the beginning to the end, an end-to-end process, which tells you basically how your process, how critical process parameters affect your quality attributes. This is what we want to have. And in order to create such a digital twin, we have to perform smart experiments. And this is something I will go over and over this presentation. I will show you a lot of details what I mean by smart experiments. So when I say smart experiments, I mean not the ordinary way how we used to perform experiments, but smart experiments mean a way how we can faster and more efficiently learn from our processes in a way that we as operators basically understand our bioprocess. So we really check, okay, how input and output is connected, and to conserve this knowledge in this digital representation. So at this level, we can say, okay, this is something like a process simulator, or it's just a modeling tool, but we want to bring this further. So once we have trained in late stage development how input and output is connected in a QBD approach, we can take this knowledge further and use it for automated process control and for automated scale up. And today I have quite some showcases for you for upstream and downstream, how this can be mastered. Before we start our journey here, I want to take you on the journey of Novasign. So quick update or quick idea for you where we are. So we started Novasign from an idea that was born in 2017 to accelerate process development by smart machine learning approaches. in 2017. Then by 2019, we founded the company and went live with our hybrid modeling toolbox. And ever then, we basically grew our company quite substantially. Who are we serving and what are our key partners? So as you see, we're already in contact, or not in contact, we have projects with quite some big biopharmaceutical companies. And on the other hand, we are partnering up with companies that basically supplement or that extend our reach. So we are partnering with companies that are, for example, in the operational field, so like contract manufacturers or development companies, but we also partner together with universities where we apply our tool to train students. And we're also partnering with other modeling companies in order to truly accelerate process development. When you think about the concept of Industry 4.0, you have to think about from different perspectives. I want to give you the angle from the digital twin perspective. So if you want to have such a digital representation of your process, the first important thing for sure is to create a plan. So when we talk about plan, I mean, like, what should your digital twin do? There are currently a lot of digital twins in the industry for different purposes, for plant engineering, for building information systems, for drug delivery. The digital twin I'm talking here is the bioprocess digital twin. So first of all, as I said, you have to create a plan. You have to create a plan what your digital twin should do. And then you have to do a risk analysis and think about which experiments you have to perform in order to train this digital twin. Once you perform this experiments, you are creating data, obviously often a lot of data. And from this data, then basically you train an algorithm. Today, I will show you some of these algorithms, how they can be trained, what they can do for you, and how these algorithms are then transferred to the digital twin to do this in silico process simulations to evaluate all of the possible process scenarios that you can think about. So what would happen if you increase the temperature? What would happen if you decrease the feeding rate, etc., to play with all of these scenarios in order to, first of all, find the optimal process conditions, and then further on to take these models for scale up and for monitoring. So when thinking about this, once you have set up your first digital twin implementation, you can then take this knowledge and plan new experiments, and then you are something like in an iterative loop. So then you can follow this loop as long as you still want to improve your digital twin, either to be more applicable for more scenarios or to optimize your process further. But once you have mastered this, once you're satisfied with the result, you can then basically, this is very similar to the slide I showed you before, then you can reuse this digital twin for scale up and automated process control. Okay, now the value proposition that we are offering from the Novasign perspective is actually here. So we help our clients to plan experiments, then we help them to execute the experiments, so we can also perform experiments for them. We are not in charge of the data itself. So here we have companies we work together with, like I mentioned before, Cubicon, for example. We also work together with Siemens, where you can acquire this data and then automatize this data transfer from their platform into our modeling platform. And then basically you're in this iterative loop. And then once you want to execute this model, or when you want to apply this model for real-time monitoring and control, you can then apply our models back to these platforms like CPAT or Cubicon in order to make this knowledge accessible in real time. Good. Now, some showcases for you that you get an idea what I mean by accelerated process development. How can you do this? The first showcase I want to show you is an upstream showcase for microbial fermentation at the 20-liter scale. And what you see here is published literature. So whenever you see a link down there, basically you can follow this link, and this is literature that we published in an open access journal. So the idea of this intensified design of experiment is faster characterization. So you want to save experiments in the lab, but we still want to get the same knowledge out. So what we do here is we have a comparison of this 20-liter bioreactor. One time we do full factorial design of experiments. So we have 3 factors at 3 levels, summing up to 27 experiments. And to the right-hand side, we were coming with this intensified design of experiment approach. The idea of intensified design of experiments is that you basically change process parameter combination during a cultivation. Okay, what does this mean? If you think of one of these lines, you see basically it starts at a certain point, then it's connected to another point, and then it's again connected to another point. And what this means is that you start with one parameter set combination in one experiment, and then over the time course, you change the parameter combination to another set point combination, and then again. So by doing so, you can visualize here that we are packing multiple parameter combination in a single experiment. In this case, we pack basically 3 parameter combinations in one experiment, so you can still screen the whole design space, but with 1/3 of the experiment. So you can think about of saving 70% of experimental effort by this approach in this particular showcase. Okay, so how does this now look like in terms of results? Or no, maybe let's go one step back. How does this now really look like in terms of experimental setup? So when you think about, normally process parameters are often kept constant. So let's say you have a temperature that is kept constant at 37 degree or 30 degree or whatever. What we would introduce with this intensified design of experiments is that we change these process parameters during a cultivation. So you can think about this orange line to be low temperature in the beginning, then high temperature in the center, and then middle temperature at the end of the cultivation. What you have to do is you have to sample over the time course of the cultivation. And what our algorithm does for you is somehow it understands the rates over time. So basically it understands what the change in temperature, how a change in temperature affects your rate in change of, for example, growing cells or productivity or certain glycosylation pattern or whatever your response variable might be. And then at the end, basically the algorithm makes use of that. How does this now look in terms of results? So what you see here is our 4 plots. So one we have once to the top left, we have a scatter plot of the biomass. And what we did here is we have these 9 experiments called training data. So we have these 9 IDOE runs, training data, and we built a model on these 9 training runs. And you see basically that this model can predict the behavior quite well. You see this basically that this diagonal line is covered. So if all points are on the diagonal line, that would basically mean that your prediction is basically the same as the measurement you're taking. And then the important thing is we take this model, which we trained on the IDOE data, and apply it to the ordinary design of experiment data, which is basically the standard data where you keep process parameters constant over the whole cultivation. And you see basically this is the test data. So there's almost 600 observations. And you can see that for most of these runs, it really performs very well. So the predictions from the IDOE on the DOE data is very high. That also means that the information that is covered in the IDOE can be directly applied on the DOE data, but with the advantage of only 30% of the experiments. So this is quite substantial. And in B, C, and D, what you see is when you take this model and then you directly apply it to these DOE runs, you see basically how the prediction of this IDOE data, of this IDOE hybrid model on the DOE data is in terms of time resolved, so from the beginning until the end of the process. So this is very, very accurate, but you can save up to 70% of experiments in the lab by applying such an IDOE concept. Here you see the same graph for the product. So before I showed you the biomass, now we have the same model predicting the biomass, the same model predicting the product. And you see there's a little bit more, it's a little bit unprecise, not that precise as for the biomass, but it's still a very good model to predict the outcome for all of these 27 different DOE runs. Okay, you can then take this model and play with it in silico. So what you can do is you can take our toolbox. Once the model is trained as a bioprocess engineer, you can go to the digital twin environment, simulate as many runs as you want in a certain design space. So here, for example, we simulated 1,000 runs in 15 seconds. And then you can play with all of these intermediate steps. So all of the intermediate induction strength here, all of the intermediate feed rates, and all of the intermediate temperatures. You can see basically how the change in these parameters would affect the response variables, in this case, biomass and product, over the whole time course of the run. What you can also do, it's not depicted or you don't see it here, you can also do multi-stages. So let's say you have a growth phase. First of all, you want to focus maximum on productivity, maximum on biomass growth. And then after induction of productivity, you can also separate this into stages. So then you would get 3 sliders per each stage. Then you can say, okay, let's please be so kind tool and find the ideal condition to yield maximum biomass in the first stage and then maximum productivity in the second stage, for example. And all of this is done in silico, just based on the initial runs that you performed. Okay, based on these findings, we then repeated the same approach in a collaboration with Boehringer Ingelheim. This is also published as for CHO cell culture, where you can basically see the same concept. So we have, in this case, two parameters, temperature and dissolved oxygen. And you can see how these parameters were varied over the time course. So while in microbial fermentations, the timing was between 14 hours, the run duration was between 14 hours and 28 hours. Here we're now dealing with 14 days. So that means it's also quite obvious if you have a cultivation that takes 14 days and you introduce something like IQE, you have the chance to screen many more conditions as if you would just continue a certain parameter set for the whole duration. This is also the reason why this was applied. Okay, and then once you have created your model, you can here again see how the model predicts basically different parameters. In this case, we were able to model the viable cell density, the glucose, and the titer here in the scatter plot. And then you can also see how this looks like in a time-resolved way. So you can basically see how the viability, the titer, the viable cells, and also the glucose was modeled over time. And because of the hybrid modeling structure, which allows you to integrate knowledge into your model, you can also see that whenever you have a bolus feed, you can model these very steep increases in glucose concentration because of the mass balances that are automatically incorporated in your hybrid model. But you can read about this in all detail in all of this publication, or you just reach out to me and I can show you. Okay, another showcase I want to show you is an improved scale-up for vaccine manufacturing. What you basically see here is also published, it's just very recent. What you see basically here is a seed optimization to reduce overall production time costs and contamination risk. And the approach we chose is when you have cells that are adherently growing, often roller bottle processes are used, where you basically scale up roller bottle production until you have the final bioreactor where you then basically transfect or infect your cells. And basically we wanted to replace this process by a beads-to-beads transfer. So the idea is basically to use TFLAS in the beginning as normal, but then go to microcarrier-based cultivation as early as possible, and then always to transfer both the cells and the microcarrier to the next stage. In order to do so and to evaluate this here, we had in situ cell detachment. So what you basically see is you see one microcarrier cultivation that was growing, and then we basically detached the cells and we attached the cells again to the same microcarriers, and we detached it and attached it and so on and so forth. And you can see basically how we were able to consecutively do this process over up to a month. So that basically tells us if we are able to regrow cells on the same beads, we can take these cells and we can take these beads and migrate them over to bigger bioreactors and to further basically grow the amount of biomass that we want to have before we basically then go into viral production. And in order to do that and to find the ideal process parameter combination, we applied again hybrid modeling. So we considered a design space which we wanted to investigate in a 1 liter bioreactor with all of the different parameter combinations, and then find ideal conditions that could be transferred along the scale to scale up. So what you see here is the result from the hybrid model that was able to describe all of these different parameter combinations in the 1 liter bioreactor. And you can see, depending on how you set your parameters, you can get up to 2.5 million cells or half a million of cells. So quite a big difference depending on how you choose your parameter set combination. This then helps you at the end to really say how, under which conditions and timing you would get which biomass concentration at which time. And this is very helpful when you then think about, okay, what would happen if I seed my bioreactor with this concentration, because maybe I have to transfer them in 2 to 4 to 5 hours, and I know when I'm transferring them in 5 hours, I would have this biomass concentration, and then when I transfer, and then when I seed the next bioreactor with this biomass concentration, where would I end up in 2 to 3 days with this concentration? And you can really start planning your process much better than right now. So based on this, then we again created the digital twin. And what you see here is a scatter plot at the end point of this 120 hour cultivation, with the difference here that each of these points in here basically reflect the true cultivation. So it's not like when you have a surface plot of a mathematical function that is built on 4 to 5 points. This at the end, what you see here is a true simulation, and each of these edges in here represent one endpoint of this cultivation. Therefore, this structure becomes much more interesting than just a quadratic plot, for example. And from that, you can basically infer which parameter combinations work best for your particular process. And then this is really important. Once you have this, you can basically we were able to really describe how the process should be scaled or should be transferred at each scale from the 1 liter bioreactor to the 1000 liter bioreactor, depending on how much volume you can transfer, what's the seeding density. This model is then really able to tell you what would be the outcome, what would you expect after 120 hours of cultivation, depending on the seeding concentration and depending on the parameter set combination you're choosing, what would come out. And therefore you can make all of the different scenarios of how long you might still need to cultivate them, etc., etc. It's a very, very powerful tool. And then in the same direction, when you think about viral vector manufacturing, so when it's then about really inspecting cells, you can then again also play with this tool. So here, for example, we have another showcase which we did together with Bio. So here you see when you then, for example, change during infection the pH or the temperature, or you can also see how the response then basically behaves. So what we see here is we can activate the cells, we can activate the virus, we can activate the double-stranded DNA that are released at the end of the cultivation, or the host cell proteins, and all of this is modeled in a time-resolved manner. So that means you don't only know what would come out at the end point, but you also know at which time point in the cultivation which level is achieved. So, and this is really important if you then think about taking this model and making an end-to-end model. So that means you take the output of this unit operation and put it into the next unit operation. So if, for example, your host cell proteins have to be purified, which is a natural process, you then directly know what's the trade-off. So the higher you basically might get in terms of productivity, you might have the drawback that you might have a higher host cell protein. And you also, as I said, know this in terms of cultivation time. So this really helps you a lot to better understand your process in a time-resolved manner. Okay, so now we are going to a downstream showcase, as I also promised. We're not only looking at an upstream process, but we're also looking at the downstream process. So here we have an ultra and diafiltration and SPTFF development. And what we want to do, we basically want to understand how a certain protein interacts with a certain membrane. This is the important thing. So we want to know how quickly this can be processed. So the idea is to perform smart experiments to screen basically a certain protein with a certain membrane. Sorry. So what you see here is with smart experimental planning and TMP scouting, we are able to predict the outcome for 4 different operational modes. So batch TFF, fed batch TFF, diafiltration, and single pass tangential flow filtration, just from the data that we receive from the scouting of a certain protein. So how does this look like? When you have this data, the algorithm, the hybrid model basically, so the machine learning part of the hybrid model can learn from this ordinary TMP scouting, which is something you're doing in the lab anyway. And then you can basically apply it to predict the outcome at each different process parameter combinations. So what we see here is we have 5 different test data. So this is now a true run, an ultrafiltration run, where we concentrate the protein solution in. And we see we have 5 different data sets. And these different data sets have differences in the concentrations, so in the amount of starting concentration, in the cross flow, velocity. So how quickly do we feed? How quickly do we feed our search tank, or our membrane? and then also differences in the TMP. And you see basically that the difference between the true measurement from the device and the model is not very large. Or in other words, our model basically looks into the future and can already tell you how long your process would take, just from knowing the current start concentration, your cross flow amount, or the cross flow feed you're basically applying to your membrane, and the TMP. So from these three parameters, the model can already tell you how long the process would last, and you can also see how accurate these models are in terms of normalized root mean square error. So we can do the same thing for fed batch. I think you have a limited amount of tank you can use. Then you normally use another surge tank on top, where you always dilute in fresh protein concentration, and therefore your kinetics is different. So due to the fact that we use a hybrid model here, we can basically just change the mechanistic part in our mass balance, and you see you get a complete different mechanistic. So while before the curve was more like convex, now we more have like a curve curve until the surge tank is empty. And once the surge tank is empty, then we really have a steep decrease in the flux, because of the fact that then the volume of the feed tank also significantly drops. Okay, you see that the error is a little bit higher than before, but still you can account for a lot of different process scenario, and you already know before you do the experiments what will come out. So this is the power of performing smart experiments like this TMP scouting, learning from smart TMP scouting, and then directly applying this knowledge for true runs in order to predict what would come out under condition XYZ. In the same direction, we can also integrate Gibbs-Dolan equation into our mechanistic part for diafiltration. So then you can also predict basically how the flux would propagate over the time course, and also how the pH, for example, would change as a function of the dia volume you're applying. The final, or the almost final thing I want to show you here is an ideal switch point. So then you can basically combine ultrafiltration, diafiltration, and ultrafiltration 2. Depending on where you switch between ultrafiltration and diafiltration, you have different timings. So the algorithm basically can predict the duration for a switch between ultrafiltration and diafiltration at 50 grams per liter, at 150 grams per liter, for example, at 110 grams per liter. So all of these scenarios can be automatically calculated to get a graph, something like this. So this you can also do experimentally in the lab, but it will cost you a lot of time and resources. Here at the end, the algorithm does it for you, and the algorithm predicts in silico where would be the ideal switch point between ultrafiltration and diafiltration. Exactly. And finally, the last thing I want to share with you here is single pass tangential flow filtration. So in the same direction, you train your membrane, or you train how your membrane and your protein interact in clever TMP scouting, and then you basically apply this knowledge for serial SPTFF or for parallel SPTFF. And when you do so, you basically see that the model that never saw SPTFF can predict the conversion rate or conversion factor for one membrane, two membranes, and three membranes, and then without performing this experiment. So since this is also published, we for sure also performed this experiment, which are these dots here. But in theory, you don't have to do this. So in theory, this tool can help you to assess, okay, would it be beneficial to do SPTFF rather than ultrafiltration in a continuous mode maybe? And what would be the conversion rate if we have a feed flow of 80 milliliters per minute, for example? So all of these questions, this process definition can be done in silico without doing expensive experiments. So because of the success, okay, here you see also for different parameter combinations, depending on how you set up your system, you see you can get all of these predictions in silico. Because of the success, we also now started to apply our tool for universities. So we are now having this digital bioprocessing at the university, at the FH Campus Wien in Vienna, where we teach students basically how to set up experiments in a clever way, how to learn from these experiments, and how to create such iterative digital twins, so that basically that groups can learn from each other in a comprehensive way, and that as a development department, let's say students would be a development department, how they can basically together optimize their process in a joint manner. So they are planning the experiments, they're executing the experiments, and they are learning how to optimize how to optimize together, how to find a certain optimum inside your process. So this is very beneficial. When I look back from my side, from how my statistics were at university, I would have loved to have something more tangible for bioprocessing. So that's why we want to also give something back. Good. With that, I'm at the end of my presentation. To sum up, 3 bullet points. You should consider the whole modeling workflow rather than just joining the battle of algorithms. I think that AI will change a lot, but you have to always think about process modeling validation. So all of this literature I showed you, we spend very lot of time to do validation of our models, what we can predict and what we cannot predict. This is very important. If you are interested in all of these technical details, just go and read these publications. It's very important, and I encourage you to do so. Second of all is if you have the combination of a smart experimental planning with a good algorithm, you can save up to 70% of experimental effort for both upstream and filtration purposes in this particular case. And finally, you can also reuse this model as I showed you with this bioreactor manufacturing to strengthen the scale-up approach. With this, I'm at the end of my presentation. I want to thank you for your time, and if you like, happy to answer all of your questions. Just reach out to me. You find my email on the internet, and yeah, have a great day. Bye.